Pleasure to have Frank here, and so maybe I'll introduce him first. So Dr. Frank BRADKE has received his PhD from Heidelberg University, and after the PhD, he moved to U.S. For postdoc fellow with Professor Mark Slovene, first as UCSF and later at Stanford. After a very brief postdoc, then he became independent, start from Max Planck, then later moved to DZNE, and together with also at a affiliate with the University of Bonn. Currently is a professor in the Faculty of Molecular Biomedicine at Bonn, and also the German Center for Neurodegenerative Disease. So Dr. BRADKE research focus on how nerve cells grow during development and how these processes can be reactivated to induce nerve injury in the injured spinal cord. So his team had a special interest in the skeleton of cell called cytoskeleton. They showed the manipulation of cytoskeleton with low dose of anti-cancer drugs led to regrowth of nerves and reduction of scarring. He actually, this kind of drug are currently has been used, right? Currently in practice in clinic, because they found a strong association between their giving gabapentin with the spinal cord. That's a huge achievement for the field. He also developed a lot of novel imaging technique, pioneered with the clearing and 3D imagings. So he's currently on the editorial board of Current Biology, Experimental Neurology, and they actually is received several quite prestigious awards, including the member of EMBO and the German National Academy of Science, and also the very prestigious Leibniz Prize, and also many others. So the first time I read your paper, I think that when I was in graduate school. I think you just finished work with Mark. So you together with others, right, you inject the second GMP to the DRG, right, to show the growth. You actually were one of the pioneer papers in the field. And also I learned so much from all the cytoskeleton. So I decided to move a little bit from cytoskeleton work on, but still axon regeneration actually. Frank is really the pioneers in the field. Okay, so without further ado, I'll give the floor to you, and welcome. Yeah.
Do you hear me like this? It's okay in the back also? Yeah? Okay. Thank you, Kai, for the very touching introduction, and also for inviting me here. This is really great. And I really have to say, for me, it was very special. Also, during the last 2 days, what I learned actually about Hong Kong, about the history, about changing perspective. Also, it was really wonderful to be with you, as well as with the students from your lab. Yeah, I think I learned so much about the history here, about Hong Kong, about China in general. Yeah, that was for me a very important visit. Yeah, so thank you all for that. And of course, I very much enjoyed the discussions already this morning. So probably everybody here in the room is really fascinated by the amazing features of the nervous system. And it's really through this complex network that we can sense our environment as well as our own body, that we can move our muscles, and sometimes even it allows us to think. And so for that, the axons have a very special morphology to wire the brain. And so they are very long, so they can reach the length of your own body, and at the same time, they're also very thin, with 1 micrometer in diameter. And just to give you a sense of the proportion, so if you would blow up an axon to the diameter here of your finger, it would actually easily reach here from the UST to the international airport of Hong Kong. Yeah, so you really have an idea how long these axons become. So we have developed more than 10 years ago actually an imaging technique that enables us also now to look on the trajectory of the whole axons within the unsectioned spinal cord. And this whole tissue technique that we have developed, that is called 3-disco, it really relies on the possibility that you can actually make tissue transparent. Yeah, you don't have to histologically section it anymore. And then if you fluorescently label single cells, individual cells, you can basically follow the cells in its entity. So the name of that technique is called 3-disco, and it has been spearheaded by Ali Ertug when he was in the lab, and then later on also followed by Andrea Tedeschi, Bert Hilton, and David Elliott. And all of these people, they further develop it in their own lab. But I think what was also very important, this gave actually a push to the field in neuroscience, and it also motivated subsequently other labs to develop their own whole tissue imaging techniques. Efforts led by the labs of Karl Dijsselroth, for example, Hiroki Ueda, as well as Carol Mason. So when I personally look on these pictures, I'm always astounded how fragile the axons look like. But if you think about it, it's actually not true. So these guys are really the tough guys because they stay with us for our entire life. And only in the occasion that people undergo a spinal cord injury or develop a neurodegenerative disease, then the axons, they actually decay and degenerate. And this is really where the trouble starts, because axons fail to regenerate. And so what you can see here is, of course, when the axons, they actually either stall at an injury site, what you see here in a mouse, where few axons are labeled with GFP. And if you find any dynamics, then it's rather that they start actually to continue their degeneration properties. However, there's a very refreshing view coming from developmental biologists, because they say, like, well, we don't understand what the problem is, because for us, actually, axons are really experts in growing and actually also regrowing and regenerating, because initially, these axons, they have to grow really rapidly to find the target. And you see here an example where actually an axon led by the growth cone goes then basically to the target. And so if you look at these points side by side, immediately becomes also clear that it says, well, wouldn't it be interesting to understand how axons grow, to really understand what are the mechanisms that make an axon to grow during development. And if we understand that really well, then we should be also able to reactivate these mechanisms under more pathological conditions, such as a spinal cord injury, to induce axons to regenerate. So interestingly, there is one point also during development where growth versus non-growth happens at the same time, and that is during a process that is called neuronal provization. So what is meant by that was actually characterized and described in a seminal paper by Carlos Dotti when he was in the lab of Gary Banker. And so shortly after the last cell division, a neuron basically looks very simple. It looks like a fried egg, not revealing anything of its later sophisticated structure. But then shortly afterwards, 4 to 5 neuroids emerge, of which one then becomes the axon. So the other one, they start to grow much later to become the axon, but of course the question is, so how does it come that basically one of these neuroids starts to grow out rapidly where the other ones are restrained from growing? And that is really an active restraint, because if you cut the axon here very short, one of the other neuroids can take over and can become the axon. So what are the mechanisms behind that? And I think there was very quickly this idea around that there's some sort of tug of war. And that means that basically each of the neuroids, so they basically pull on the cell body, and the one that basically pulls strongest becomes then the axon. And the idea behind it was that maybe this sort of pulling mechanism that basically could here enable the growth cones that are here in these processes could pull basically the whole axon behind it. This was based on a mechanism that has been actually characterized and defined by Tim Mitchell and Mark Kirschner many years ago when they described how fibroblasts actually move forward. And so, you know, these fibroblasts, they have here the actin filaments, and they have this typical treadmilling, right? So actin is basically inserted here at the plus end, it moves them backwards, and they're depolymerized at the minus end. And so this sort of treadmilling that then goes on, once it is actually used through a clutch mechanism, once basically it is mediated to the extracellular matrix through adhesion sites, that actually then enables here to move the leading edge forward and thereby also pulls the rest of the cell behind it, right? This is called pulling, or also the clutch hypothesis. So people thought maybe a growth cone is nothing else than a fibroblast on the leash that basically pulls the axon behind. However, it became also clear that this cannot be the whole story, because already in a very beautiful review from Mark BRADKE, he actually said, well, neurons, they can actually grow out their neurons without actin filaments, namely when you treat them with cytokeratin D. And I thought really one of these remarkable studies came from Paul Forscher and Stephen Smith when they described actually what is happening when you depolymerize the actin cytoskeleton of Aplysia growth cones. What you see here by Nomarsky microscopy are the microtubules that basically form the backbone of the neuroid shaft, and they are surrounded by a very dense meshwork of actin filaments. And now let's see what is happening when you treat them with cytokeratins, and you see that very quickly the actin filaments around here, they depolymerize, and instead now the microtubules protrude very quickly into the area formerly occupied by the actin filaments. And so if you think about it, also it was a very drastic perspective, a change in perspective compared to this pulling, right? Because for the pulling, you need the actin, myosin, you need basically contraction, similar like what we do also with the muscles. Whereas here it seemed rather that the actin cytoskeleton actually restrains the microtubules from protruding. So if you think now about the polarization, maybe what is happening is not that actually the growth cones are actually pulling along the substrate through the actin network, but maybe normally the non-growing neurites, there's a very strong actin restraints, not allowing the neurites to grow. And only in the future axons now, there's so much actin dynamics, the actin becomes loose, and therefore the microtubules can actually now protrude and the neurons start to polarize. So if this is true, one would expect what is happening if one gets rid of the actin filaments in all of the different neuroids, right? So instead of just having now a single axon where you basically create space where the microtubules can go in, so you would have now actually also microtubules growing into these other areas, right? Instead of having now a single axon, what normally neuronal polarization makes, right? The neurons normally have just a single axon, so you end up with a cell that actually has now multiple axons to start to grow. So these data, they gave really the first basis now to understand how on the level of the mechanism now neuronal polarization can occur. And subsequently, many different labs were actually interested in understanding the signaling upstream that basically then triggers down to the cytoskeleton to direct neuronal polarization, including the lab of Kozo Kaibuchi, as well as Andreas Püschel from Peleu, as well as Josh Sainz. So personally, I was actually, after this time, I was very interested in another question. That was like, maybe this, what we understand here, could also help us to understand how growth and growth sustained is actually also regulated after a spinal cord injury and may even lead to axon regeneration. So I think you're all aware, and I think also due to the beautiful work from Kai, what is happening after spinal cord injury. So there are many inhibitory factors in the spinal cord that prevent injured axons from regrowing. And one idea for a long time is that one tries to basically neutralize some of these inhibitory components, including factors that are called LNOGO or chondroitin sulfate proteoglycan, as well as OMGP, for example. However, what I thought at the time was that maybe it would be possible to understand actually the brake inside the neuron that actually keeps the axon on hold, and where ultimately many of these signaling components converge on. And I believed actually that the brake that is acting here has quite something to do with a brake that restrains the growth of the other neurites. So, of course, you would not also just like to understand this process, but you also have the possibility to manipulate, to loosen the brake, and therefore to elicit the axons to regenerate. But obviously, this is not like a one-person's job, but for this, you really need a team effort. This is really the premise of our lab was built on such a team effort to understand now how these axons during neuronal development, or so how we can basically employ these mechanisms to elicit axons to regenerate. So the first thing that we were doing now is to look again onto the cytoskeleton. So I've been telling you that basically the actin filaments, they restrain the microtubules from growing. And you could also imagine there something like a fisherman's net, that basically where the microtubules cannot poke through. But maybe the question is, are the microtubules just passive regulated, or could they basically play also a more active role in this process? So could they basically push their way here through the actin network? And indeed, that was the case. So the microtubules are more stable in the future axon. But Harald Witte could also show more. So this is here basically a longevity marker for the microtubules using acetylated microtubules compared to the tyrosinate ones. But apart from this correlation, so Harald Witte also showed causality. When you use now a low dose of a microtubule stabilizing drug, and I really stress here low dose, so it is basically 10 to 50-fold lower than what you would have normally in a anti-cancer treatment, where you normally would freeze microtubules altogether. But by using these low doses, you still enable that the microtubules can actually grow, but they do not shrink that often anymore. And as a result, you get these more stable and pushing microtubules. So here again, so you end up more with multiple axons, and it therefore determines these microtubules, these stable microtubules, this initial polarization. But that is not all. It also creates something like a landmark for the cell where the axon was. And you remember what I told you before, so if you cut here the axon very short, then one of the other neurites can take over, whereas if you cut further distally, leaving some of these stable microtubules intact here, that this can be used as a nucleation seed from where the axon starts to grow out again. And so what Susana Gomez-Ruiz actually showed is that actually this is not only working in the young neurons, but even in fully mature neurons that are integrated into synapses. And if you think about it, it's a quite a drastic scheme of neural plasticity. So when we normally talk about neural plasticity, what we mean is that basically you have a strengthening of certain synapses. But if you challenge the system here very drastically by cutting the axon, you can even change the function input to an output and vice versa. So it's a bit like, imagine like changing the projector to the keyboard or so on. Yeah? You can really transform input and output. So we knew now that basically the axons, they seem to grow, but actually not through the actin filaments, basically by pulling on the matrix. It seems rather the microtubules seem to push. And so it means that probably this idea that a growth company pulls to make an axons to extend longer seems to be not quite the case. But to directly test this, now Thelma Santos and Barbara Shafran, they actually cultured neurons in a three-dimensional matrices. And what you can now see in these different matrices is the following. So when you culture fibroblast, what you see is that they actually pull on this matrix, exactly as Tim Mitchelson and Mark Hirschner was saying. It looks a bit like a spider web, where they basically go the whole time in and out. But by contrast, if you look now onto the neurons, there's nothing of these pulling also. You cannot see any of these pulling, but actually the neurites and the axons, they grow without any of the deformation of the collagen fibers that you can see here so effectively by the fibroblast. So we knew now microtubules rather seem to basically push. These axons, they grow in this amoeboid movement, and you can actually even transform an axon to a dendrite and vice versa. But of course the question is, so how come that you cannot reactivate this polarization program in vivo after a spinal cord injury. And so here, Ali Ertug and Jörg Rüschel, they looked now what is happening after an injury in the central nervous system where axons fail to regenerate, and they looked to what is going on after an injury in the peripheral nervous system where axon actually can regenerate. And they used for asking this question the so-called dorsal ganglion neurons. And they're a really beautiful system for that, because they actually have an axon in the peripheral nervous system, so that if you cut this one can regenerate. And they also have an axon in the central nervous system that fails to regenerate when you normally cut it here in the central part. And so they looked for very different parts. They looked for energy supply, they looked for membrane traffic, but they did not find there much of a difference. But where they did see a drastic difference was again on the level of the microtubules. So after an injury in the peripheral nervous system, where axons can regenerate, microtubules remain assembled, they remain bundled, and they can actually create a pushing force that the axon can start to grow out again. By contrast, after an injury in the central nervous system, the microtubules disassemble, and the few ones that are left, they basically just grow in any sort of direction. So these ones, they cannot create any pushing force to make an axons to grow out here. And this is something we did not find only in the rodents, but actually also Jörg and Ali found it also in spinal cord injured patients that lived for more than 4 decades with a spinal cord injury. So very differently to neurodegenerative diseases, so after a spinal cord injury, the axons actually persist at the injury site. But what you can also see here, the microtubules, they remain disassembled. So in a way, that was good news and bad news. And the good news was, well, it seems to be quite a specific effect, because it really affects only the microtubules. And you can actually transform also these growing axons into non-growing retraction bulbs by microtubule stabilization. However, it was also bad news, because normally the microtubules, once gone, they would have to be reassembled at the so-called microtubule organizing center, the MTOC, which is located at the centrosome. And that is quite a daunting task if you think about it, because so these axons, they can be, of course, like a meter long, and if they would have to be formed from the centrosome here at the cell body, they would have to be transported for a very long time. So quite a daunting and challenging task for a little neuron. However, one has to say that much of the knowledge about what is going on with the microtubule nucleation and in cell biology stems from the work on mitotic cells. And obviously, neurons are post-mitotic, and we therefore thought it would be worthwhile to look into more detail again. And indeed, what Michael Sties found is that the centrosome becomes actually inactivated as a microtubule organizing center during neuronal differentiation. And so the good news in a way was that now the microtubules, they're assembled not at the centrosome, but actually they can be formed in the axon, in the cell body, as well as in the dendrites. And actually, only recently, we could actually show that even the polarization, so the initial axon growth, happens completely without centrosomal microtubule nucleation. But instead, the centrosomal microtubule nucleation is very important for the migration of the neurons. And they actually show a very beautiful ballet that is actually occurring in the brain. And so, I don't know whether you're aware of also, but actually neurons, so basically the cortical neurons, what is happening to them is that they normally move up to the cortical plate. But at the same time, out of the trailing edge, they have the axon that starts to grow out here, and basically that goes into the other direction. So you have a very sophisticated ballet, imagine that. So you go up on the one side, but on the other side, basically the axon comes out. And so what now Stanislav and Sebastian could show is that by using the centrosomal microtubule nucleation, you can disentangle these two processes, these two dynamical processes, because the centrosomal microtubule nucleation becomes switched off once basically the neurons actually reach the cortical plate, but the axon here can continue to grow, and also it can even grow then here onto the other side, to the cortex callosum, but without any centrosomal microtubule nucleation. Yeah, that does it completely centrosomally. Okay, so in a way that was really good news, because it meant that you don't need centrosomal microtubule nucleation, but the axons, they simply can just get replenishment of the microtubules within the axon itself. And actually, Mika Spira, he showed that in aplysia neurons also that you even get some sort of microtubule organizing center after an injury. So that was good, and so we thought like maybe it's therefore worth to use now microtubule stabilizing drugs to elicit axons to regenerate. But there was something else what is very interesting when you use Taxol and other microtubule stabilizing drugs, because they're used in the clinic not only as an anti-cancer agent, but also as an anti-scarring agent. And so if people, for example, receive a coronary stent, this stent is usually coated with a low dose of Taxol, and simply if you add the stent into the coronary system and you would actually have a small mechanical damage, this of course would be detrimental for the patient, right? But using the Taxol, you actually abolish this scarring event. And why this is important, we come now to the point, because when we ask the question, what are actually the minimal requirements for axon regeneration? So to ask this question, I would like to reintroduce to you the DRG neurons you just heard about. So remember, there's this peripheral axon that can regenerate, and there is a central axon that normally cannot regenerate. However, Richard and Issa, they showed in a seminal paper, if you first cut the peripheral axon and subsequently you cut also the central one, you actually have regeneration. And I could show together with Simona Neumann that you can actually mimic this effect also by stimulation of the cyclic AMP pathway. So it was really nice. However, it does not work in the clinically relevant timeline. So you start basically here the treatment always before you have the injury. And so, of course, that doesn't make sense. It would be a bit like you want to ride your motorbike and then your partner says, but honey, don't forget your cyclic AMP injections also, right? It wouldn't make much of a sense. So what is the reason for that? So why do you need to do it basically before? And in principle, in biology, there could be two reasons for that. And the first one is a cell intrinsic problem. So you could imagine that after an injury, after such a CNS lesion, these axons here atrophy, and even if you afterwards perform this peripheral lesion, that the neurons, they cannot acquire growth competence anymore, and they cannot start to regenerate. The cell extrinsic concept would say something different. They would say like, well, after the injury, of course, there are still not growth competence. You could trigger growth competence with a peripheral lesion, but something has changed so drastically at the lesion site in the meantime, namely a scar, for example, is formed, that even this growth competent axons cannot cross the scar anymore. So to distinguish between these possibilities now, so Pavna Ilera and Ali Ertug, they made many different experiments. And I just want to show you here the key experiment that they were doing, because what they performed now is simply a lesion to cut an axon without creating a scar. That is something what you cannot do mechanically, right? Because whenever you, even if you take the finest scissor, you always would have there also some scar tissue around. And so instead, what now here Pavna Ilera and Ali Ertug was doing, they performed a 2-photon laser lesion. So they cut a single axon and then waited for a couple of days and then performed a peripheral lesion. And what they indeed found is now that the regeneration occurs. Yeah? And so that means that really the scar was the major problem for the growth competent neurons. And I show you here now an example of these sort of experiments, the raw data basically. So you see here a mouse where few axons are labeled with a GFP signal. And what you see now is this axon here is now cut with a 2-photon laser, and it actually starts to degenerate within hours. The one that is directly below that one, that was not attached because this is a 2-photon laser. What you see here now with the injury, so this is where the cut was. If you don't condition them, if they are not growth competent, they actually stay at the injury site, even if you don't have a scar. So you have a little bit of arborization, but not much growth. If you afterwards then perform this perfilation, if you cut the perfil axons, what you see now is that within a couple of days, these axons here start to grow and to regenerate robustly. So what this data therefore tell you is, of course, this is not a way how you heal spinal cord injury, but it gives you a biological concept about what you need to do to enable axons to regenerate. So you have, on the one hand, you have to reduce the scar, but on the other side, what you also need is to get the neurons in a growth competent mode. So you need the cell intrinsic mechanism and you have to reduce the scarring. And I think now becomes also clear why we were so excited about using now Taxol, because in principle, Taxol could do these two things at once. It could reactivate the growth state and it could reduce the scarring. And I will show you now two short video sequences that should basically illustrate the mechanism. I will show you first a neuron cultured on this inhibitory substrate, on a substrate that is called NOGO-A, one of these inhibitory factors. And you will see now the microtubules, how they polymerize. You won't see the whole microtubules, otherwise basically it would be, the whole video would be just white, it would be bleached. But what you see is just the polymerizing ends, the tips of these microtubules that is labeled with a microtubule end binding protein 3, or also called EB3, fused to the fluorophore cherry red. So initially what you see is that you see the satellites just going everywhere. Yeah, so they're basically flowing around, showing that microtubules polymerize and then they shrink again. If you add now 2 hours of either Taxol or another microtubule stabilizing drug that is called epoxyalon, what you see is now that the microtubules, they grow and polymerize to the very tips. Yeah, remember, you only see the tips. And if you look after another 2 hours, what you find now is that these microtubule tips, they actually push now these neurites forward. Yeah, so basically they start to grow by these microtubules, pushing them forward. And if you basically look at for hours and for days or so, you see then also how much they extend. So here you reactivate the growth state, but at the same time you actually abrogate the polarization of the so-called scar-forming cells, and this is the reason that they cannot move into a scar. Yeah, they cannot basically go to a lesion site. And you probably have heard from your biology, cell biology 101 lessons that normally fibroblasts move in a directed fashion because they basically have stable microtubules in the direction where they grow. But now imagine you use a microtubule stabilizing drug. You have now in the front, in the back, left, right, you have everywhere stable microtubules. So they still can wriggle around, but they cannot actually move in a directed fashion. And so what is happening now, if you basically create here an injury site, an artificial injury, so whereas the control cells, they move very quickly into this area formerly occupied. So you see that the epoxyalon B treated meningeal fibroblast, so they wiggle around, but they cannot basically close it in a directional fashion. So these data, they had been also then actually recapitulated by other labs. So for example, the lab of Phil Popovich showed this reduction of the fibrotic scar. The lab of Dietmar Fischer actually could also show that by this low dose of Taxol, you actually increase also now optic nerve regeneration. And something what I was in particular excited was a paper from the Jin lab. So she works in San Diego on C. elegans. And what she found is that when she looked for mutants of neurons that cannot regenerate, normally the worm can regenerate the axons really happily, but she created now very different mutants also that fail to regenerate. But when she fed these worms now with a microtubule stabilizing drug Taxol at low doses, that actually enabled them to regenerate. And I think what it makes so interesting is that it basically means that we are tapping here into a very conserved mechanism that is conserved throughout the animal kingdom. So these two things, basically axon growth and disrupting of the migration and thereby also the deposition of scar-forming factors such as the CSPGs. This together enabled now axon regeneration and functional recovery. And I think one thing what I really also would like here to stress that this is a process that also works in stroke. So in a very nice collaboration we made with the lab of Gabor Patzelt. So here's actually a clinician, but also a scientist. And so we looked there in preclinical models for stroke or so, and that also enabled now for axon regeneration and function recovery. So the way how we tested them was to test now the animals treated with epoxylon and their walking ability. We use here a moderate contusion injury. And what you will see now in this moderate contused animal, so this is a control, is even though they would walk normally on a flat surface pretty well, so they basically make here mistakes with their back paws. So they basically go through the grid here, and you see the chock, how often they basically fall through. So they make these mistakes, whereas now animals that were treated with epoxylon, in this case the epoxylon D, they actually move over in a very coordinated fashion. So we were at that time already very excited about the data, and we shared them early on prior to publication with other labs, including the lab of also including the lab of did I show you both of the videos? Aha, okay, now then, sorry, then so wait a minute, so here, so this is a vehicle one, making the mistakes. And now the EPO-D. It basically moves over without it. Fine, both seen? Okay. And so basically, we early on shared our data now with two different labs, namely the lab of Armin Blesch and Norbert Weidner. And they found also in a complete different rat strain, namely in the Fisher rats, they also found a similar function improvement. So we are of course now very excited about these data, and we will also continue now to study them in more detail. But also using, for example, combinatorial trials, so using other drugs, and on top of that, also to test that in, for example, in a cervical injury model. So these are experiments that are currently ongoing. But what I would like to stress here is now also that we are interested in really understanding the molecular mechanism underlying axon regeneration. And so we think that we would be now in a position that we can really decipher the different mechanisms by really going a similar strategy as we had before. And remember how we came actually to the microtubule stabilization in the first place, and that was actually that we used now the neuronal polarization as a model. And exactly this is what we are doing also here. So we now deciphered first neuronal polarization and to have basically entry points to then get into the axon regeneration. And so the strategy was straightforward. So we are asking always three questions. So the first one is, we genetically ablated candidate polarity genes in the nervous system. And so, for example, here's a row JTPase, the C42, and what you can see is they fail to form an axon as what you would see here in the wild type. So this is a physiological knockout. What we did then is in this physiological knockout, we looked for the relevant downstream effectors. So in this case, for example, from RAG1, we found that the wave regulatory complex is actually not where it was supposed to be. And what we did then as number 3 is that to really see is it a linear cascade, we restored axon growth by actually overexpression polarity mutants and looking now for the manipulation of this downstream effector. And here's one piece or so where we use basically the microtubule binding domain of the CLIP170. And by going through this round of different research strategies, we could actually identify now a very systematic way how neuronal polarization occurs. That was experiments driven by Dorothee Neukirchen, by Bojan Gavalos, Sabina Tahirovic, and Sebastian Dupas. And so I won't give you now all of the details of all the different signaling that is in there, But I think what is important as a take-home message is that there's nothing like a canonical signaling pathway. So it's not just going through one specific cascade, but what comes out that they all converge on either making the actin more dynamic, more loose, so that the microtubules can actually protrude further distally, or that you stabilize the microtubules and thereby basically push their way through the actin network. Basically, the interaction between the microtubules and the actin also is something what we are currently heavily pursuing. So, what does it tell us now, having this knowledge about how axon regeneration also could function? And I really would stress here, of course, this is a question of neurodevelopment that is exciting per se, but of course we really wanted to do now the next step. And let's now zoom in for this onto this molecule, cofulin. So what Kevin Flynn showed during his postdoctoral time is that actually cofulin is very important for the transformation of this more fried egg phenotype of the cell when they start to transform into a neuroid-bearing neuron. And so the way how it does it is that actually here the actin filaments, and you remember about this actin retrograde flow that I was telling you, that here it severs the actin filaments and thereby creating space where the microtubules actually can move in. And you see that also in the video. So in green you see all the actin filaments, you see all the dynamics, but if you wait for a little moment or so, you see that where formerly now the actin was, actually actin is depleted, and instead now the microtubules started to move in. So cofulin is used to dynamize the actin cytoskeleton to create space so where microtubules can actually go in. But in the adult nervous system, what we found is that actually the actin cytoskeleton is very rigid. And so thereby it does not create any plasticity where normally the microtubules can actually go in. And so we therefore thought like maybe by overexpression of cofulin in the adult nervous system, that would be then sufficient to induce axons to regenerate. Yeah, and in a very heroic effort from Barbara Shafran, Michele Cuccio, Andrea Tedeschi, Sebastian Dupas, as well as Claudia Laskowski, they overexpressed now cofulin in the adult nervous system in injured neurons. And what they found is now that these axons actually cross the injury site and they actually regenerated. Yeah? And you see here this typical tortuous structure of a regenerating axon. So we therefore thought like, well, maybe we would be now even in the moment or so in the position that we can decipher a whole signaling cascade that normally would suppress axon regeneration. And remember what I told you, that there are many inhibitory factors in the spinal cord that prevent the axon from regeneration. We talked now about the microtubules, a big deal, and also how the actin filaments restrains actually the microtubules from protruding by forming this compact actin network. And so Sina Stern and Brett Hilton therefore went into the question, so what could be really the whole signaling cascade going from the outside to the inside or so, and they're restraining actin compaction and this microtubule protrusion. And so we have learned actually quite a lot nowadays about the different ligands that are inhibitory. And here I mentioned a few. There are also now the receptors known. And what was very interesting was that there's actually a node, namely the ROA, a signaling pathway, that seemed to play an important role. But you see in this review some years ago, they did not know much what is going on downstream. So 6 to 7 years later, actually they learned quite a lot more about the signaling processes, basically the receptors, the ligands, but again, there was not much knowledge what is going on downstream. And so we thought, therefore, it's really important to understand what is going on in this downstream. And that is not just an academic question, but it also had a clinical relevance because Lisa McCarricker, in her beautiful work, she actually used a toxin, a bacterial toxin, to inactivate ROA and to induce axon regeneration. That has been used in clinical trial by Michael Fellings. However, these trials were put on hold because the patients were not doing any better than the patients that were not treated with the C3 exoenzyme, with this bacterial toxin. And we thought like it's even more important that we understand the process that is really going on there. And so one thing what I really would like to stress is, of course, we always like to zoom in of our favorite cell type, right? But of course, the nervous system does not exist just in neurons or so, but there are also many other cell types in there. And so, therefore, what now Sina was doing is to create now a knockout, first of all, where the neurons are actually ablated of row A. And what she found is that when you treat now wild-type cells with inhibitory factors such as CSPGs, you find this compaction here of the actin filaments, and you see how the microtubules are restricted. This depends on row A, because in the row A knockout, what you find is now that the actin filaments stay where they are, and also the microtubules stay where they are. Yeah, very different here in the wild type, yeah, where you get this actin compaction and also the strain of the microtubules. So therefore suggested that we have now here a cascade where the row A compacts the F-actin, and thereby the microtubules become inhibited from further protrusion, thereby inhibiting axon growth and regeneration. So, but what could be the molecule that basically compacts the actin cytoskeleton? Well, we learned through Sebastian Drepras that during development at least, there myosin 2 forms together with actin a network, the so-called actomyosin corset, that restrains here the microtubules from protruding. So there's something like a corset or so where the microtubules would protrude. And this is very important during development because basically the way how the neurons grow is not in a continuous way, but they basically grow, but then they stop again, and then they look for any guidance molecules, and then they start to grow again, then they stop, and then they sense again for guidance molecules. So you need some pausing in between. And this pausing is actually mediated through row A and the actomyosin complex, yeah, through this myosin 2. And indeed, what now Sina found is that actually row A plays a very similar role in the adult nervous system. So actually ablation of row A is something very good, because then the microtubules could in principle protrude, these axons could start to grow. However, if you ablate row A, not just the neurons, but also the whole nervous system, you have actually a counter effect for regeneration. Namely, when you ablate row A in the astrocytes, these ones become hyperreactive. So you get actually here a gliosis, and that basically neutralizes the pro-regenerative effect by the row A ablation of the neurons. So what you therefore would like to do is that you either ablate row A only in the neurons, for example by gene therapeutic approach, but not in the astrocytes. Or what you also could do is that you go downstream, yeah, that you simply see where are the signaling cascades downstream of row A diverge in astrocytes and neurons. And where you do find this difference again is on the level of the microtubules. There's simply 50-fold more microtubules in the neuron in comparison to the astrocytes. Yeah, there are very few, and they can basically, by the stabilization, you could overcome here now this effect. So I show you now these data, also the raw data, by using this whole tissue imaging approach. This is how it looks after spinal cord injury. The injured axons, they stop at the injury site. You get a very similar effect if you ablate row A, both in the neurons, but also in the astrocytes, right? So basically stop, yeah? And so you actually have the beneficial effect of row A ablation in the neurons, but then this counter beneficial effect in the astrocyte. And this one becomes clear if you ablate row A only in the astrocyte, because we even find now these U-turns where basically these axons actually start to go away from the injury site. And only when row A is ablated in the neurons, you find now that these axons actually cross the injury site and you find axons to regenerate. So what is therefore important is really when you think about signaling, and if you want to look for manipulation to elicit axon regeneration, is that you also have to see like, well, you are actually basically dealing with an ecosystem, a cellular ecosystem. You cannot just focus on the neurons, but you also have to see what sort of interaction we have with non-neuronal cells, and how can you manipulate them in a way that you actually get them axon regeneration. So this in vivo cell biology, so I therefore consider very important. So what I would like to do now at the end is to ask actually a very simple question also, because the whole time we were saying like, well, in the adult, the axons, they fail to regenerate, they lose this intrinsic property to grow, to regenerate, but they are so good in growing in the juvenile form during development. And of course the question is, so what is actually the reason for that? Why do the neurons lose actually their growth and regenerative competence? What is the reason for that? And of course in science we normally don't ask the why questions, but I really like it also because it helps us to conceptualize. So where could be the points, where could be the issues? And you can imagine like a very simple scenario, and that is that actually during development, the axons have to rapidly grow. But once they have found the target, they should form synapses. And if they would continue to grow, they would actually do some misrouting, and they would make misconnection, and therefore leading then also to misinformation. So at that moment when they make synapses, they should actually downregulate their growth competence. And if you have then an injury, then these axons, they should remain actually growth incompetent. So if such a hypothesis is true, then you would expect that there would be a growth suppression program that is initially not there. That initially the axons, they grow, but once synapses are formed, that this growth suppression program becomes upregulated, and it remains also upregulated when the CNS axon is injured. But is there any evidence for such a growth suppression program? And actually there came a very nice introduction from a paper from the late Ben Barres, and he actually said that electric stimulation actually slows down sensory neurons. And here this is the original paper from the Cater lab, where they actually studied sensory snail neurons, and you see when you stimulate them with electric activity, they actually stop to grow. And Joanna Enes showed the very same thing also in adult DRD neurons. If you culture them, they actually stop to grow during this growth. And actually conversely, Bert Hilton showed if he performed a CNS injury, and I think one of the students was asking me at lunchtime also this question. So if you actually here now cut the axon and you pharmacologically silence them using a DRADKE approach, they actually start to grow and to regenerate. So remember what I told you, like during electric activity, there should be some sort of maybe a growth suppressor, also a growth suppressing program is upregulated. And I think one evidence came from Joanna Enes that really suggested that something like this happens. So what Joanna was doing, remember, if you electrically stimulate, they stop to grow, and she used here the depolarization using KCL. But when she performed these sort of stimulation with KCL in the presence of transcription inhibitors, so she basically suppressed now transcription, what she found is that these axons continue to happily grow. And that actually suggests that indeed, by this electric stimulation, there could be a transcription of a growth suppressor. And so to search for such a growth suppressor, Andrea Tedeschi performed now the following experiment. So he used for 3 different experimental paradigms, where either actually neurons are in a growth competent or growth incompetent mode. So in total, this was actually 9 different procedures. And then he looked using a bioinformatic approach. So what are basically, could there be something like a common program? And out of a complex bioinformatic analysis, he came up with 5 genes, of which one was CAGNA2D2 that encodes for the voltage-gated calcium channel subunit alpha-2 delta-2. That was actually very interesting because alpha-2 delta-2 is a synaptogenic protein. So the late Ben Barres, in fact, Charla Eroglu, when she was in his lab, showed that alpha-2 delta-2 is synaptogenic. And so here we are now at this moment. We are looking for something that is involved in synapse formation and that could also then block actually axon regeneration. So interestingly, alpha-2 delta-2 you can actually block by gabapentinoids such as pregabalin and gabapentin. And now when Andrea treated animals after spinal cord injury with pregabalin, he found anatomical regeneration. And he subsequently also showed later in his own lab that these animals functionally improved after spinal cord injury or even a stroke in rodents. And actually, again, Gabor Petzol's lab showed in a very nice collaboration very similar result for the stroke. So I will show you now in a moment whether that might have also some clinical implication also for the users of gabapentinoids such as pregabalin in the clinics. But remember, I think we are here really at the initial beginning also to understand how axon growth and synapse formation, how they're basically singing to each other. So how is it that basically either you make the axons to grow and then synapse formation should be actually put on hold, or you should basically have the synapses formed or so, and then the axon growth should be on hold. And the first step to a better understanding came now by Brett Hilton when he actually manipulated the synaptic vesicle machinery. And what he found is that if he used now an ablation of the vesicle priming machinery, he actually induced the axons to regenerate. And he could recapitulate these effects also now by pharmacological manipulation, namely using baclofen, another drug that is actually also in the clinic. Okay, so we have now microtubule stabilizing drugs that are in the clinic. We have the baclofen and we have the pregabalin. So could this be something of interest for the clinics? So we still don't know, so we don't have any clinical data. But early on, we actually shared our data with the EMSCI network. This is a center of 20 spinal cord injury centers across Europe. And the beauty of this consortium is that they actually look onto the patients with the same test everywhere. And so they have basically a very big data chart of thousands of patients. When they performed a meta-analysis of the data, what they found that actually patients that were, for other reasons, on pregabalin or gabapentin within the first months after a spinal cord injury, they were doing better functionally. And a similar result also occurred when patients were treated with baclofen. So this is by no means now any clinical trial or so on, yeah? But this meta-analysis gave them enough hope that they actually also changed now the standard operation procedure of care that was now introduced. So patient with a spinal cord injury, they will be delivered into this EMSCI network. They directly started to be treated with pregabalin, yeah? So, and to see now what sort of effect that will also have for their potential function recovery. So, and again, here it is important to stress this cannot be a one-person's job. It can also not a team, just one team or so, but to really ask this question, you need a whole consortium. You need a team of teams. And to really to see that we can eventually translate this finding in a potential therapy. So what I would like to offer you now, instead of a summary where we just go on step by step through or so on, I would just like to remind you, I think you guys are clever enough, so I don't think you need a summary, but I just would like to remind you how we came to that one, and that was that we wanted to understand how neurons grow during development. Remember, these guys are experts in growing, and so the more important is to understand, to dissect the mechanisms that maintain axons to grow, and to employ these mechanisms to induce axon regeneration in the adult. And this actually, having said, I really would like to stress that it doesn't mean that by no means we have understood even neuronal development yet. And I think that one came to me as quite a surprise out when a student in our lab, he made some basically very funny experiments. So we thought like, well, this is not happening. But by this way, he transformed completely our thinking of how about the microtubular structure. So I used to think about the microtubules basically like pillars in a room, right? So they're basically very static, and maybe something above and down. So this is where they polymerize. So what he actually showed that these pillars that basically are important for the structure of the cells, they are constantly moving in a retrograde way. Yeah, it would be like the pillars here that you have here in the room would be just moving constantly down, but the ceiling is not changing because at the same time already small brick stones are basically put on top of it. Yeah, something it basically overturned the dogma that is around for the last 30 years. But there are many other very important questions that are currently asked or so on, yeah, and very important question. And I really would like also to stress we still don't understand actually how axons grow in vivo. Yeah, so far we understood them in cell culture. We made now also quite a step forward where we analyzed how axons actually grow in three-dimensional substrate, but to really understand how they grow in vivo, we actually still don't understand. Yeah, and I think with this, I think we are now at the end, and I just want to give you here a teaser. It seems that we are also awaiting in the moment a paradigm shift. And remember what I told you at the beginning, so people thought for the last 30 years that basically you have this pulling from the growth cones or so that here each of them wants to become the axon to grow out very distally. But actually Tianchen Lin, with a very fantastic work, he actually just showed now that he overturned actually this growth cone-centered view, and he actually shows that a lot of this polarization is actually here going on at the cell body. Yeah, basically by a very stochastic, very interesting mechanism, and we believe that actually this mechanism that he actually here identified, and I'm happy to tell you more also if you have time, but that will also very important to transform our knowledge how an axon is regenerating. And maybe it's not happening so much actually at the injury site itself, but also again, certain directionalities also from the cell body. But with this, I really would like to finish now by thanking all of the members of the lab. As I stressed before, this is a team effort, and I was very fortunate that I have very good people in our lab. The present one, but also the past one, of which many of them actually now themselves team leaders, group leaders, professors, or even directors of whole research institutes. And it's really nice to see how they're growing. Yeah, and finally, and I think this is also something what I usually also like to stress. So we had a meeting, I think, when I won one of these prizes. So I think the first thing what I did with the money is to invite everybody back to Bonn. The present one, the alumni, so on, and everybody was telling what they were doing. And it was nice to see that, of course, the ones in academia, they were super happy. But it was also nice to see that actually the people that left academia, they were also happy. They found their job in industry or in the government or so on, and they gave also to us very exciting perspectives. So I think what I try to say is whatever you do, and just follow your heart, and I think you will find there a good way. I'd like to thank all of our collaborators that helped us a lot with much of the heavy lifting. And I'd like to thank you for your attention. And these are the funding sources also for and I'm very thankful to the funding agencies also for their generous funding. And I thank you for your attention.
Thank you. Actually, very comprehensive work. I mean, I think myself, maybe I'm also a student, got very impressed. What I learned from you is how to, you need to ask the right question. Right? I mean, this, I hope this is going to be inspiring to many of the students here. Okay, questions from the...
Not everybody at once.
Yeah, maybe I can just start. You know, because I also work on your activity-induced growth.
Yeah.
At the time, we tested BRADKE. BRADKE and DRG inhibit exactly as you showed. But the question is, central neurons, peripheral neurons, why? Although you think it's only because of the receptor express difference, or there are clearly some more fundamental difference between the two types of neurons. Yeah.
So I give you my speculation. It doesn't mean that it has to be true. Yeah, but I would argue that DRG neurons are often very different for many reasons. So also developmentally, they come from the neural crest. It's not that they come basically where normally the CNS neurons come from. And there's a special occasion also where the signals come from. And so basically, if you think in this way, so my drawing is really bad. So if you basically the question is, if you cut here, you make it growth competence, and what could it be? Yeah. And there have been some ideas with immune cells and so on, but I thought there is a very beautiful paper that I'm sure your students all know. It's the paper from Smith and Skeen. Yeah, so if you don't know it, it's something you guys really should read. It's the Bible. It's two molecules. Yeah. And this one, the Smith and Skeen paper is from '95. But what they did is, in principle, to take the DRG neurons out. And what they found, if you just let them wait in culture, also they become growth competent, arguing that probably the immune response is not the most important thing to get them in a growth competent mode. Yeah, but I'm happy there also we can also speculate in a different way. But based on this paper, so we actually thought that maybe basically there could be a negative signal that basically, as long as you have the connection here to the target, that basically suppresses axon regeneration or axon growth, because it basically says something, don't grow, you are still connected. Yeah. And if you have now the injury, this signal that comes here along is not getting there anymore. So if that is true, it may be, you could then also argue that maybe electric activity is the point, right? Because the muscle spindles, so the DRG neurons, the proprioceptive one, they have the muscle spindles here, and you have the whole time the electric activity. If you cut here, so what simply means is then basically this electric activity that silences the growth mode would basically not keeping them. By contrast, if you don't make it here, but if you cut here, so the electric activity would still go to the cell body and thereby basically keep basically the suppression program working, right? Whereas if you cut here, this signal would not come here. And the same if you first cut here and afterwards cut here, so then also the electric activity would be here down. And so the idea could be therefore, so coming to your question also, that basically this electric activity is basically this growth suppression in the DRG neurons. I would be far from arguing that something like this could also work for other CNS neurons. So that has to be tested. And yeah, we have to see. This is in the moment, it should be actually off the record or so on. But I thought that this is not happening in hippocampal neurons. It looks now in the moment or so, even there also you have some growth suppression. But we still have to understand, is it some sort of toxicity actually there, or is it really like a physiological signal? Yeah? So we are starting now but that will take a couple of years, but they use then, for example, also optogenetic tools also to actually stimulate them. Yeah, but we are slow, so we don't know yet.
Okay, great talk, great work. So I just very impressed, your Axon regeneration after you overexpress the coffee.
Yeah.
Coffee is simply just an acting regulator. I'm wondering, the underlying mechanism, about the underlying mechanism. So it's possible actually simply stabilize or the stimulator cytoskeleton actually make an axon pushes through this actually the injury site and give the momentum for axon regeneration. Alternatively, could also maybe help to actually activate cross-clamp, which actually help overcome as a barrier formed by injury site.
So you mean, sorry, I did not understand everything also, but you basically mean that also in degeneration process that
No, actually the overexpression coffee, coffee overexpression, it promoted the axon regeneration.
Can you say again? So you understood it, right?
Coffee.
Yeah.
Overexpression promotes axon regeneration, right? You present data.
Yeah.
So because then what it actually does, because it's just regular locally, right? So what exactly this does to help axon?
Sorry. Yeah. So basically, what we did is also so we have our cell culture assay, and what we did also show there that basically with the cofilin, so you basically enhance the actin turnover. So if you have them on inhibitory factors, there would be basically not much growth. If you add now cofilin, you basically enhance it, and basically thereby you have them grow out again and so on. Yeah, that was basically the primary thing. Whereas it is also necessary and sufficient, because if we performed the conditioning and we knocked out cofilin, then they stop to grow.
Somehow they have to change, the physiology of the neuron in order to for regeneration, and not just just change the actin dynamic in the tip of the axon. Right?
I think it's very important what you say. I think I just enjoy it.
Because when you overexpress coffee,
yep.
Although you're saying this looks like only axon has growth tip, right?
Yep.
But King's point, you might still need something going on with the neuron, right?
So cell body in order to
side, also the cell.
Yes, absolutely. That can very well be. And so I cannot exclude, for example, with the cofilin, there's also, or they're basically the SRF, they're basically a transcriptional component that could be also changed or so on. Yeah, that one we cannot completely rule out. That is true. Is this what you wanted to say?
Yes.
Yeah. That is. No.
Dynamics, in tip.
No. I think there you're. Yeah, there you're right. Yeah.
Thank you. Lovely talk. I enjoyed it a lot. I'm confused about something. So you mentioned the example of where you use the same cell, but is it the same cell? One side goes to the CNS, and if you cut that, it doesn't grow back. With the PNS, if you cut it, it does grow back. Right. So, and then later you asked the question of why, and you postulated that perhaps it was because once it reaches the target, it no longer needs to.
Yeah.
But that would be true of the other side as well. So what is the sort of evolutionary motivation on the PNS side to maintain the ability to regenerate then?
Yeah, absolutely. So it's a question I have to say we are asking also that in the lab, and I cannot give you a right answer to that. Yeah.
Okay, so maybe to follow up with that question, when you talked about the tip and the microtubule and actin behavior, I immediately thought about cell polarity and the biophysics of that. And then you, at the end, you mentioned the stochastic process that you're doing. Could that be related? Somehow establishing the cell polarity on the PNS side versus CNS side somehow confers different ability to basically extend the structure in one direction or another.
So I think from what you're saying, I think the point that you're bring up here is well taken. And I would argue, so during development, it's relatively simple because they start to grow out, and apparently, at least for the DRG neurons, so there's papers from the 80s that actually show that in the peripheral nervous system and the central nervous system, they reach at the same time the target. Yeah. So from this side, we are actually fine. But I think I don't want to discuss away the point that you're making. So I also think there's in the moment a hole in our argumentation. Yeah, because what you're saying is, so with the synapse formation in the CNS makes sense, but why? What is going on in the PNS? And I honestly, I would be cautious to try just to argue against it, because I think it may be that to understand this one will be crucial for the next development or so on. Yeah.
Okay, thank you.
Yep.
Other questions?
Thank you for the interesting talk. So about alpha 2 delta 2 part, so I want to ask maybe two questions. First is kind of general. So that totally makes sense in terms of network integrity. So once network is built by targeting to specific locus by forming synapse,
Yeah.
then they should stop, circuit formation. So without those systems, maybe additional synapse or additional brain region will be targeted by different axons. So if the neural activity dependent gene expression suppress further axon generation, then if you inhibit those pathways, I would expect not only regeneration is accelerated, but extra circuit formation will be built.
Yeah. Yeah.
Do you think so?
Yeah. So that is that you basically would have some sprouting also. That could very well be. So the fact is that in the intact network, it doesn't happen. And I think one reason for that is, so for example, pregabalin is given to people that basically, if you have a very hard pain with your teeth, yeah, then even you are getting the pregabalin. But it's not that you basically lose your memory by that. However, if your network is still not rigid, then it basically becomes more loose. Yeah, and so for example, during development, so I would. I was also talking with the students about it. So when Andrea made the experiments in the embryo, yeah, so basically giving pregabalin through the mother. So what he found is now that basically there was less synapses, and basically the axons, they were also continue to grow. Yeah, and so basically you keep basically there some growth and so, and you mess up the network. Yeah, it's consistent with the idea. Yeah. Yeah. But in a rigid network, so there, basically, I doubt that is possible. So to change it or so on, but you really need also the hit. So you could also argue in a very similar way if you think about the taxol, right? I mean, one thing that we were at the beginning afraid with, we felt like, if we give microtubule stabilizing drugs, maybe we would now also in vivo get also cells with more than one axons, right? Which of course would create a chaos in the nervous system. And that is not the case, so you don't find it. And I think in the moment, so that is a fact. Our explanation and our speculation is that in principle, if the system is intact, you also often have like something what is called the perineuronal net around it that basically keeps the nervous system and the network in check, yeah, that it's basically fixed. Yeah.
Maybe we actually have a receptions at the 5th floor lab science. So maybe you can talk more about that. Okay, great. So let's thank Frank again. So give a fantastic talk.